So the lecture today will be about a short introduction into deep learning in general. So I will give a short introduction into convolutional neural networks. I will give a short introduction into LSTMs because we will need both techniques in order to understand a new way to get to distributed vector representations of protein sequences. And for this we will use a technique which was published for natural language processing roughly a year ago and has since then revolutionized the field of natural language processing, and we are by now hoping to bring this knowledge also to the field of bioinformatics. And at the end of the talk I will also give a short introduction into state-of-the-art architectures for secondary structure prediction, and yes, that's it. So let's start right away. So first of all, a short introduction regarding the motivation and the background, how we actually came to the point that we would like to use a different vector representation than it was used for the last 20 years. So first of all, I think you know already this figure on the left over here. So it just boils down the fact that proteins which share a certain sequence similarity also share structural similarity, and this structural similarity also always implies functional similarity, and by looking for similar proteins we also harvest evolutionary information from these proteins. So you can understand this is a kind of data augmentation if you're from the field of computer vision, for example. And for example, at this point we see that we need roughly the number of residues which should be aligned should be 100, and so we need roughly 40 of 100 residues to be aligned to be above this curve and to actually be able to benefit from or to assume that two proteins share the same structure. But this is just the background why we actually would like to search for very similar proteins in our machine learning workflow. So here on the right I've just shortly sketched a rough outline of a usual machine learning workflow in bioinformatics. So I've skipped some parts like the redundancy reduction and the cross-validation splits, but just in general you always have a query sequence, a protein sequence here, and you always start by searching for this query sequence in a huge reference database. So this can be UniProt, this can be uniclust, and this is usually in the range between 15 million sequences and 2.5 billion sequences. And there you already should notice, okay, fair enough, this might take some time, and yes, this actually takes some time, and you have to repeat this process for every protein again and again. After you've searched for very similar proteins, you can align these very similar proteins by introducing gaps into the proteins which you found to be similar to a certain sequence similarity level. And from these, from this multiple sequence alignment, you can then calculate a matrix. So basically the vector representation which was used in bioinformatics for basically the last 20 to 30 years, which is called position-specific scoring matrix. I think you should by now be familiar with this, as the last exercise was also covering this. Or the next one will be, not 100% sure. But basically what this shows you is a matrix of shape L by 20, and L is just the length of the protein, and 20 is basically just a score which indicates how likely it is that a certain amino acid at this position how likely it is to observe this amino acid at this position. What you actually see in there are the log odds probabilities, but you can just understand it as a matrix which shows you probabilities, how likely it is to observe a certain amino acid at the position in a protein. And given this matrix, you usually start training some type of artificial intelligence, so some kind of neural network, convolutional neural network, LSTM, whatever you come up with, and then you come to the inference point, and then you, for example, here just do a secondary structure prediction in 3 states. So you always try to predict 3 classes, let it be helix, sheet, or coil, and then you can do a one-to-one translation from this query sequence to this 3-state secondary structure prediction which you then induce. And yes, that's just what was done until now, basically, and what is still mostly used. But again, the time-consuming problematic part is here, that you have to search for every protein sequence, again, these huge databases. So again, just to recap, what was the problem about this previous approach, which was used like for the last 20 to 30 years? Again, it's computationally expensive, as you have to search the reference database, which contains millions of other sequences again and again for every protein. And also, it's not applicable to all proteins. So you can imagine that there are certain cases where you simply do not find any similar proteins. So proteins which are intrinsically disordered, for example, are just harder to align, and therefore you will find less evolutionary related proteins, and therefore the information in your PSSM will also be lower. And the predictions based on this PSSM will be of lower quality as well. Then proteins of the dark proteome. So these are just proteins which are not which are either hard to crystallize, which have less interest by biotechnology companies, for example. So we simply do not know much about these proteins for several reasons. But the fact is just that we do not have any homologous sequence which has an annotated structure for these proteins. And therefore we will again have only a small multiple sequence alignment and a less informative PSSMs for these proteins. Here, down in this graph, I'm just showing the development of the database sizes. I think this is quite small to read, but it's not necessary. It's just showing different databases which contain protein sequences. So basically, databases which can be used as reference databases over here. And you see how the number of sequences in these reference databases increases exponentially over the last years, basically. And with this, also the computational time, of course, increases as you have to search these reference databases again and again, always, for every new protein you would like to make predictions for. And yes, this is just very time consuming. So what did actually others do? So in the field of bioinformatics, it was usually limited to harness this evolutionary information by, again, just looking for evolutionary related proteins, create multiple sequence alignments, and so on. But in the field of natural language processing, it's not possible to align sentences and deduce something like a substitution score for specific positions in a sentence. But what did they do in order to get a distributed vector representation for words? And I think some of you might already know this figure. It's from the word-to-vec. It is an example for the word-to-vec algorithm. It was one of the first algorithms which did in an unsupervised way, which retrieved vector representations for words or strings in an unsupervised way. So before this, you always had these statistical methods like the inverse document frequency stuff, where you rather counted occurrences and related them to the expected frequency to observe them. But the word-to-vec was one of the first algorithms which actually did this via a neural network and in a completely unsupervised fashion. And by doing this, they achieved something where you can represent words like man or king or queen or women by a vector, and then you can represent these words in a vector space. And now the interesting thing was that the relationships between these words had actually a meaning. So you could now do math with words, basically. You first represent the words with vectors, and now you can suddenly do math with these vectors which represent words. And for example, here on the right-hand side, you see a PCA transformation of the vectors for these words on the left and the words on the right. And you see that there is a certain relation between the countries and the capitals of these countries. So the neural network found a vector representation which actually understands certain relations between words. And again, this is not like magic or something like this. This is just something like statistical properties, so it's just more likely that Spain and Madrid will appear together in a sentence than, for example, Spain and Hanoi. So it's not something like, like magic. It's just doing the statistical analysis, which was done beforehand, basically by hand, automatically in an unsupervised way. And this is the difference. And now we would like to apply a similar approach to protein sequences, which can be basically also interpreted as words in a vector space. But first of all, before I explain now the natural language processing technique which we applied, I will just shortly recap CNNs and LSTMs because I was not sure how many of you are actually familiar with them. So can you just raise your hands if you have worked with CNNs or LSTMs before? Okay, perfect. So this makes it a lot easier. So, and if you find any mistakes in there, just let me know, because maybe you already know more than I do. So first of all, let's just start with a short comparison between the normal feed-forward neural networks, or just neural networks, and convolutional neural networks. So for the normal neural networks, which were used basically before 2011, something like this, you always had a fixed input size, input layer, and then you had several hidden layers, and you just fully connected all of the input nodes with all of the nodes in the hidden layers, and then you got a prediction at the end. In contrast to this, CNNs now allow you to use flexible input size, and the neurons can be arranged in 3D. So there you already see that this was basically invented in the field of computer vision, but by now basically all other fields started to adopt this, because it does not need to be arranged in 3D. It's just something you can have, but it's not that you have to have this property. And then every layer transforms the 3D input. Again, does not necessarily have to be, but can be. The 3D input is then transformed to a 3D output. Okay, this is a very superficial way of looking at it. So now let's look a little bit deeper into this, because it might be hard to understand actually. So here you go from the input. This is again an example from computer vision. So you have here image 32 by 32, and then you have these three channels, the RBG channels, and then here is just the convolutional kernel or filter depicted, and then you just move this filter step by step over the image, and then you simply do a dot product with the underlying, so between the underlying values and your current filter kernel, and by doing this you just produce a new feature map, which now represents your image in a higher dimensional feature space. So in this feature space now you could have, for example, something like edges which are detected in the underlying image. And every of these kernels just detects a different type of feature. So one could detect edges, other convolutional kernels could detect round things, something like this. So you can just build up more complex features by going deeper. So the first layers will only detect very simple aspects in your image, and then deeper layers will start to combine these easier features which you predicted previously. And down here you see a relation to the actual, the intuition behind this, why it's also still called neural network, so where it originated from, so the idea of the neurons in the brain. So here you get an input via the axon from another neuron, so this is our X0, so this would be one of the values in our image, so this would be just, for example, the red channel in our image, and then we have here weight W0, so this is the trained weight in this filter kernel up here, and then you simply multiply these two guys. In biology, you would propagate this signal via the dendrit, and in the cell body, the actual function happens. So there you do this dot product basically, and then you add a bias, and of course we also need this activation function. And this is also similar to neurons in the brain, because there is also a certain threshold which needs to be reached until a neuron fires. So this can be related to this activation function. And the output is then basically just, again, this activation function, which is in usual cases just a simple rectified linear unit or a sigmoid function. Then you simply multiply every value in the image here with every weight in the filter, sum it up, and then you add a bias. And then you move one step to the right, for example. So in this you do repeatedly for the whole image. And at the end you get a feature representation of your whole image when you propagated the filter kernel over the image. Here on the right you see a more fine-grained example. So this is your input. Basically the input just goes from 1 to -3, and we have here now a convolutional filter of size 3. And in order to get a valid convolution, so to produce an output which has the same size as the input, you need to do zero padding, so you just add 1 0 to the left and 1 0 to the right, and then if you apply this filter you will get an output which has the same shape as the input. And then you just basically move this filter kernel of shape 3 step by step over this input, and then you produce by multiplying and summing up the output values. And that's basically the whole idea behind CNNs. And now let's just talk about the advantages of CNNs, and maybe also already related to protein sequences. So first of all, the disadvantage of normal fully connected neural networks is always that the receptive field is limited to the window size which you've chosen. And deeper layers will have absolutely no information beyond this window. And this is an obvious limitation, so you have always fixed input sizes. So if your image would now not have the shape 32 by 32 by 3, but for example 33 by 32 by 3, then this algorithm would already break, because you would need additional input nodes which you would have to train again. And this is obviously not good. So in some fields this might actually work. So for easy toy examples like MNIST, you could safely assume that you always have the same input size. So you could absolutely train the whole MNIST toy data set where you just had to classify handwritten digits via a normal fully connected neural network. But again, this is a toy example. In real life, your inputs will have different shapes. And also the problem would be if you would just basically flatten the MNIST data set and just put pixel next to each pixel and then feed it into a normal neural network, you would have to learn certain features again and again. So there would not be one filter which just learns what an edge looks like, but you would have to learn these features again and again in different neurons in your input. So the advantage of CNNs is by moving these filters over the input, that you have these shared parameters, and they are translational invariant. So this means that no matter where you observe an edge in your image, you will always detect it by simply moving the same kernel over the image. So it doesn't matter where or in our field, for example, where the helix is. We could also design a normal feed-forward neural network which has size 100, and we would always feed fragments of proteins with size 100 in there, but then the neurons would have to, or some neurons would have to learn always again to detect helices, for example. But this doesn't make sense. You always need just one convolutional filter which knows what a helix looks like, and then you just move it over the sequence. And this is already all implemented automatically when you use convolutional neural networks. Another advantage is that the receptive field, so the number of residues which you take into account when you make a prediction, grows with the number of layers. So the deeper you go, the bigger your receptive field becomes, and as well as with the window size of each convolution. So I think it is obvious that if you use a kernel of this shape, 3 by 1, you only have a receptive field in the first layer by one neighbor to the left, one neighbor to the right. But you can, of course, extend this and look at 2 neighbors to the left, 2 neighbors to the right, for example. And now also let's relate this maybe shortly to the neural network which Professor Rost published. So here you would have had the sequence to structure layer in the first feed-forward neural network hidden layer. And in the next layer, you would have this structure-to-structure hidden layer. But actually, these were two separately trained neural networks, as far as I know. And this idea basically resembles convolutional neural networks. So this is something which you have to keep in mind. So this was like 20 years ago, and by splitting here this into two different neural networks, so basically you have one input layer, one hidden layer, and then you have an output. You do all the predictions for the output layer, and then you train another neural network on top of the output of the previous neural network. So this resembles somehow the power of CNNs to create intermediate feature maps, and yeah, basically to create intermediate feature maps, and then use these feature maps again to make predictions. The only huge difference there is by doing this split here, that you could not do an end-to-end training, which is obviously not perfect. So after you trained the first part of the neural network, the weights stay fixed and you cannot change them anymore. So the second network has to take whatever the first network has already learned. But by doing this in this end-to-end trained fashion, in like CNNs do, you will have the advantage that the first layers can also change their weights again. And now let's look at an example which basically resembles this idea of a sequence-to-structure layer and a structure-to-structure layer in CNNs. So you would move, in this case again, just a 3x1 kernel over some protein sequence which just runs down here. Again, we padded zeros here to have a valid convolution and to get an output of the same size as the input. I just omitted the padded elements here and here to make it less overloaded. And then you just move this kernel down the sequence, along the sequence. In this case, I've omitted the feature channel because basically you would represent every amino acid, which is here, not by one value, but by some kind of a vector. So you would have, for example, in the previous case, always a vector of size 20, if you would use the PSSMs. And in the later case, which I will show you, we would have a distributed vector representation of size 1024 to represent every of these amino acids. And then you just move this kernel along the sequence, and then you get an intermediate intermediate feature representation of this input, and then you do the same with another kernel, which already takes the pre-processed features, so something which already relates to structure, and basically you just repeat this process. And then you get to the final prediction. And yes, the advantage obviously is that no fixed input is required. So again, here for normal neural networks you would have fixed input, and for CNNs it doesn't matter whether you have a protein sequence of length 10 or 1000, it will just move the filter a little bit longer over the sequence, that's it. But the algorithm wouldn't break if you would add some residues. So this was just a short introduction into CNNs, and now let's talk shortly about LSTMs. So in order to understand LSTMs, you also need to understand, at least on a superficial level, the normal recurrent neural network, which are a little bit older than LSTMs. So what you're doing up here is basically you have an input x at the time point t, and you want to make predictions for this time point called ht. And as I've already mentioned, the time point, so you have sequential data, so depending on your background, you can either see it or understand it as protein sequences, they are ordered sequentially, or in the field of natural language processing, you can understand it as a sentence which is built up of several words, or if you have rather an economics background, you can understand it as stock market prices. So here you take the stock market price today, and then you want to predict the stock market price for tomorrow or something like this. And you do this sequential, or you introduce this inherent sequential bias into the model by introducing loops, which do not only take the information from today into account, but for the stock market prices, for example, they also look at the price yesterday. And then they make a prediction about tomorrow by using the data from yesterday and today. And that's it. And now we have introduced this loopy structure here, and you can also unroll this loopy structure when you start at a certain time point x0. So in protein bioinformatics, this would be the first residue. In natural language processing, this would be the first word in a sentence, or basically the time at which the company actually sold the first shares of her company. Yeah. And then you can simply always absorb one of these data points, no matter. So let's just stick to the protein example. So the first residue is absorbed, and then you make a prediction about the first residue. You go on to the next residue. You absorb the next residue, but also consume information from the previous residue. And now you make a prediction about the second residue while looking at the previous one and the current one. And down here we're looking into these small boxes here, what's actually happening in here, and you already see that basically they are just concatenating always the current input with the previous information. And based on this and some nonlinearity, so in this case the tan h, they do a prediction. So this was one of the first approaches to actually model sequential data. However, the problem of this, or yeah, I think this is the better slide to explain this. So this works perfectly if you have like 10 data points. This should work without problem. But in real life, you usually have longer sentences, you have longer protein sequences, and your stock market prices do not only go back to yesterday, but like 20 years ago. And then suddenly you run into problems because your recurrent neural network keeps forgetting about the first part of the information while it tries to make predictions about tomorrow, for example. So you cannot look back in time like 20 years by these recurrent neural networks, and therefore the long short-term memory was introduced, or in short LSTMs, and as you can already see, they became a little bit more complicated, but we will just look at every single step by step. Here is just a small legend. Every yellowish box is a neural network layer. Then we have pointwise operations where we simply add or multiply stuff. We simply transfer vectors from one place to the other. We concatenate them or copy them. And yes, so why are LSTMs actually better at modeling long-range dependencies than a recurrent neural network? So obviously they get more complex, but introducing more complexity doesn't necessarily lead to better results. But what they did here is they introduced something like the cell state, which is just carried on from LSTM, so from every time point of the LSTM to every other time point. And it only gets minimal edits, so it is only modified minimally. So you only forget some of the previous stuff, and then you just add some of the new stuff. But you only minimally modify the cell state, and this allows the gradient to easily backflow during training, or to backflow more easily during training. Exactly, so information is only added or removed, and this can be understood as some kind of a conveyor belt which processes information between time points. And but how do we now add new information, or how do we actually forget some of the past information which might not be needed anymore? So for this we have in the first place what is called a forget gate. So the formula is up here, but basically you're just concatenating the new input information with the previous hidden state of the previous LSTM. And so you're concatenating it, and then you're simply running a sigmoid transformation which squeezes the values between 0 and 1, and these values can be understood as some kind of a weighting of the new information. So values closer to 0 are rather forgotten, and values closer to 1 are kept. So they are the important ones. Yeah. And the forget gate, yeah, just chooses how much information we want to forget or keep. in a next step, we have to also add new information. So we do not only want to forget something about the past, but we also want to add new information about our current stealth or about the current residue which we've absorbed or the current stock market prices. And what we do here, again, we just absorb the new information. We take the previous hidden state, we concatenate them. We again run only a normal hidden layer with a sigmoid transformation in order to assign weights. So this just says what information we want to keep from the new input, so what we actually want to update. Again, these are just values between 0 and 1. 0, again, this is unnecessary information which we do not need to add to the cell state, and values closer to 1 indicate this is important to make the next prediction, so that's what we want to keep. And the same vector goes in here and receives a tan-h transformation, so this is just a normal hidden layer again with a different nonlinearity, so these values do not need to be now between 0 and 1, and these are actually the values which are our feature representation, if you want to say so. So this is our new cell state, which we now want to update. And yeah, this is our new candidate cell state, exactly. So now we have just prepared the updates, but we did not perform the updates. And as easy as this, so the forget gate simply again just contains values between 0 and 1, and you simply multiply them with the previous cell state. This allows you to kind of forget some of the previous information and or keep, depending on what is important, and vice versa. Over here we're assigning this weight which is stored in IT, and we multiply it with our new candidate cell state in order to only add some relevant information but not all of the information. And then we simply sum the previous cell state, which has already forgotten the stuff which we do not need anymore, and we add the new information to the vector. And now we've modified our cell state, but we also want to make predictions based on this, and therefore we need to create a new hidden state. And this is again done by just using the concatenation of the current input and the previous hidden state. We again just run a normal neural network with a sigmoid, just squeeze it again in between 0 and 1, and then we do a tan-h transformation of our new cell state. So at this time point, what is already received here as an input is already the new cell state. So this already has forgotten stuff about the past, and it was already updated with new stuff. And now we run here actually the nonlinearity, because otherwise the new cell state wouldn't have received any nonlinear transformation yet. And then we simply multiply it again with this new information. So this is then our output. So this is necessary because we have two different channels, as you can see. So we have this conway belt over here, which just updates by forgetting and adding new information, and we have this hidden state down here, which then is actually affected by all these transformations, which you see down here. Okay. So this was just a short introduction into convolutional neural networks and LSTMs, and what do we need them for now? So this was kind of complicated, but now I would say the fun part actually starts, because now we apply this stuff also. So again, what did others do to get this distributed vector representation? Because this is where we want to get to. We just want to go away from PSSMs because they are hard to compute and they do not provide enough information for all proteins. So this was our motivation why we went there. And other people did via this Word2Vec algorithm already this natural language processing stuff. And how did they actually get there? So the Word2Vec algorithm was already introduced a few years ago, and the problem of the Word2Vec algorithm is that it will treat the same word always with the same vector representation. So there is no context dependency, and you can immediately understand that there are some words in every language which are just ambiguous. So you use them in different contexts, and then they have different meanings, and this is something which you would like to represent by this vector representation. representations, and this was not possible by word2vec. No matter in which context you use Madrid, it will always have had the same vector representation. And I think Madrid might be a bad example because this will always relate to some kind of a capital. But if you think about, for example, Apple. Apple can be either a fruit or a product, which you can, or a electronic device which you can buy. And there you certainly need different vector representations, which was not possible by using word2vec algorithm. But now, by using these so-called language models, you allow to actually use contextualized information. So the same word will have different vector representation depending on the sentence in which it stands. So depending on whether apple is now in a sentence where a tree occurs, or in which I don't know, a laptop or a smartphone occurs, you will have different vector representations. But what did they actually do? So they took the whole Wikipedia they took as a training corpus, and then they basically just moved with LSTMs over every sentence, and they tried to predict the very next word in a sentence given all previous words. So I think here in this example is visualized quite good. So we have this sentence, let's stick to improvisation in this skit. And now what the model, what they called Elmo, does is that it just absorbs word by word by word and tries to predict the next word in the sentence. And this is done in order to allow unsupervised training. So usually it is quite expensive and time consuming to get labels for your data. This is a job which no one wants to do, so that's why they started to harness the information from Wikipedia right away. So you do not need any labels for this. This is the huge advantage. You basically just take an input sentence, feed it to the neural network, and then you just, to get the targets which you want to predict, you just shift all inputs one step to the right, or to the right. So whatever, in one direction. So, but basically you're just always predicting the next word. So that's the whole point here, and you do not need any labels. And so this is just one LSTM layer moving in the right direction, and they did not only use one LSTM layer, but they stacked two LSTM layers. And what is happening down here is a so-called character CNN, and this character CNN allows you to squeeze words of different sizes into one vector representation, because as you can imagine, in the field of natural language processing you have always words which have different numbers of characters, but you want to get one vector representation for every word. And this is a perfect use case for CNNs, because no matter how long the word is, you just move CNN over all characters in this word, and then you combine these the outputs of these character CNNs, for example, by max pooling or by summing, by multiplying. But just the important thing is that you come up with a single vector representation of words, no matter how long they are. And as you can imagine, for LSTMs, so some of you already know them, you do not only move them in one direction, but usually it's useful to use them in a bidirectional fashion. So you move them from the left to the right and from the right to the left. And correspondingly, you always try to predict the next word in a sentence, or if you go from the backward model, then the previous one, basically. And yeah, this is not important, just how to get to the actual embedding. So what we extract here are then the hidden states. And here is just what we did. So we took the Elmo model, which was used in natural language processing. We trained it on a huge corpus which contained protein sequences, and we considered every single amino acid in a protein sequence as a single word. So we always observed amino acid by amino acid and tried to predict the next amino acid in this protein sequence. And as you can already see here, this is something which you obviously have to do on GPUs, because on CPUs this would not be feasible. And even on 5 GPUs, this took us 2 to 3 weeks to somewhat converge. So it's, you can always train it longer, but we didn't see any huge improvement there anymore after 2 to 3 weeks. Okay, so now we have trained this model. We get vector representations for amino acids out of this model. But what do we do with them? Because the natural language processing model just spits out performance called perplexity, and this just indicates the certainty of the model about predicting the next word. But this is not something which allows us to compare to what other people did in bioinformatics. So again, just a short recap. So this was the previous situation. Again, you search for a sequence in this reference database. This took all a while, then aligning PSSMs. You train some AI. Now, in our case, what we did is we just plugged in the new protein sequence into the already pre-trained ELMO model, and then we got embeddings out of them. So these are now these contextualized distributed vector representations for protein sequences, and you see already the matrix has changed. So previously we had L by 20, which were the log odds probabilities for how likely it is to observe amino acids, and now we have 1024 features which are describing the amino acids at each position. You might wonder why 1024? It's the default. So you could also use 512 or 2048 probably, but as it took 2 to 3 weeks to train this thing, we did not experiment heavily with this hyperparameter. But basically you could also choose something else. And now we had this vector representation of all the proteins in our data set. Now we wanted to train some kind of a network on top of this. Yes?
1024, which dimension is that in the matrix? Or is that the whole...
So which dimension is this? So you have in... If you stick to this example, then you would have in the columns always, or over the columns runs the sequence, and every amino acid in this sequence is then described by a vector of size 1024. So I'm not sure what you mean by which dimension, because you could also just flip the dimensions in this case. This doesn't matter. It's just like you describe every single amino acid by a vector of size 1024. Previously it was 20. So you can already imagine that this, of course, causes some problems, or might cause some problems if you have really small data sets. So if you have a data set of 300 proteins and you have already an embedding size of 1024, then this might lead to problems. This is an obvious disadvantage of this approach. But again, this is a hyperparameter which could be chosen differently. But now we would like to compare to what other people did, and we would like therefore to use secondary structure prediction as a kind of a proxy to assess the performance of our now pre-trained language model. And therefore we came up with a very simple architecture depicted here on the left. We kept it as simple as possible to rather see the information content which is stored in these embeddings, and not to basically create just a super complicated architecture which by itself would already produce reasonable results. So therefore we kept it simple. As the input, we basically just used this matrix, so it has shape L by 1024. Then we train 32 different convolution kernels with window size of 7 on top of this input. So this means we're always looking 3 neighbors to the left, 3 neighbors to the right, and based on these neighbors we then create a intermediate feature representation. We also use some dropout as regularization. We use rectified linear unit as nonlinearity, and then we do not only single class prediction, but we have a multi-class prediction problem here. So we want to have secondary structure predicted in 3 classes, in 8 classes, and we also want to predict whether a residue might be disordered or not. And just regarding 3 and 8 classes, so for 3 classes you have helices, sheets, and basically everything else. So the 3rd class is just a summary of whatever doesn't fit into the first 2 classes, and the 8 classes are just more fine-grained, so you have different flavors of helices depending on the number of amino acids which are required to do one turn, for example. But I will also talk about this in a few slides. And we do a binary classification of whether a given residue in a protein sequence is disordered or not. Yes, here. So maybe some of you are also familiar with something which is called seq-to-seq. So this is something which is heavily used by now for translation, for example. So you're basically just translating a given input sentence into a given output sentence. In most languages, you have the problem that that you do not have a one-to-one relation between the English language and the French corresponding sentence, for example. In our case, this can be mapped to a seq-to-seq problem, but there you have a one-to-one relation between the amino acids in a protein and the three possible labels, again, helix, sheet, and coil, or whatever. And here you see the other states, so G, H, and I are just different flavors of helices, EB are just different types of sheets, basically, and STC are just coily, loopy, whatever. And disorder, yeah, just binary classification, is it disordered or not? The definition in this data set was that you do not observe the residue in the X-ray crystallography structure, so it's just based on missing experimental results, and then you claim that it's disordered. So this definition has also some shortcomings. What I found out during working on this project, this might also be important if you plan to work on something related in the future. If you want to compare to other people's work, watch out, there are different mappings from going from 8 classes to 3 classes. And as you can imagine that as soon as you use a different mapping here, then you cannot compare the results at all anymore. And yes, there are also different definitions for disorder. So these are some of the pitfalls you might encounter when you compare your work to other people's work. Here is just more visualized view of how these CNNs actually process this information. So convolutional kernels are always shown with these colors. So basically only these two guys here and here, and this and this, or only these two are intermediate results, or basically this is the input. So this is again our embedding input, so this is a matrix shape L by 1024, again feature representation created by Elmo, and then you just move this convolution which is shown down here. This is a convolution of size 7, so you have, you're looking at 3 neighbors to the left, 3 neighbors to the right, and this is the center residue. And at these corner cases, you need to pad some elements in order to have a valid convolution, so these are indicated as red here. But as soon as you would move now 1 step to the right, only 2 of these guys would be red remaining, because you only need to pad the corner cases. And then you simply move this filter kernel over the sequence step by step. You do a convolution between this convolution kernel and the underlying matrix. You do not want to do this only once, but you do it 32 times. And by doing this, you create an intermediate matrix which has shape L by 32. So you create an matrix which still has, of course, the same number of residues as the original matrix had, but now you have 32 feature channels, 1 feature channel for every convolutional kernel which you've trained down here. And then you do the same up here, but there you do it only once, because in the final layer you already want to have a prediction for our secondary structure, and therefore we train only a single last convolutional layer which already produces our output probabilities. So then let's look at a performance comparison to state-of-the-art approaches. So we have 3 different test sets, CASP12, TS115, and CB513. It doesn't matter, it's just like different test sets looking at different aspects and focusing on different things. And the reference here is NetSurf P2, and this is currently considered as one of the state-of-the-art methods, and they reach Q3, so accuracy in 3 states for 3-state secondary structure prediction of roughly 86%. And here is NetSurf P1, the previous version. You see a huge improvement between these two, and there are just different other methods. So Spider 3, Raptor X, and JPRED 4 are just different other methods out there which already utilize deep learning and PSSMs to make also 3-state prediction. But they are all using evolutionary information harnessed from PSSMs. And here is now our approach, Seqvec, so you can already see that there is still a huge gap between the state of the art and our current approach. But you also have to keep in mind that the pipeline which they deploy, okay, I would have to jump back many steps, but basically what I want to show you is just a slide where you see that they had to calculate MSAs and PSSMs again. So in our case, our prediction does take only 0.01 seconds or something like this, so you can do it on the fly. And in their case, this only creating these multiple sequence alignments can already take minutes. And then you can imagine how long it takes to annotate, for example, the whole human proteome with 20,000 proteins if a single protein already has an average processing time of several minutes. And we could make predictions for the whole human proteome in like half an hour. So this is definitely the advantage. So you basically do a trade-off between speed to performance. And yeah, we're working on closing this gap now. So to be fast and accurate. And we did not apply it only to this use case, so secondary structure prediction was our first proxy to see whether the trained embeddings actually work. And the second proxy was a more global point of view on proteins. So now we are not looking at every single residue in a protein and say whether it's a helix or something else, but now we want to say whether the whole protein is rather membrane bound or rather soluble. And for this we have used DeepLock dataset. Forgot to put the reference here, I will add this later. But basically what they had is a data set of roughly 8,000 proteins, and every protein in there had a label which said it is membrane bound or not. And in order to get now a fixed size vector representation of our proteins, we did the same like what natural language processing guys do. They simply sum up over all the embeddings in a given sentence and then calculate the average. So in our case, we simply summed all the distributed vector representations in our protein over dimension L. So we collapse dimension L by simply summing all the vectors. This results in a single vector of size 1024, and then we also divide by the length, because obviously you will get different values whether the protein has here only length 10 or length 1000. And then we basically just represented the resulting 1024 vector. So every protein was now represented by a fixed size vector, and this fixed size vector was now broken down into 2 dimensions using the t-SNE dimensionality reduction. And you can already see that the raw embeddings without any pre-training, so we never told the model actually what a membrane-bound protein looks like and what a soluble protein looks like. You can already see that without any of this information, that there is down here a nice cluster which clusters membrane-bound proteins, and other clusters, okay, here you always have some noise in there, but again, this is without any pre-training or any notion of what a membrane-bound protein looks like. This is just the result of basically predicting always the next amino acid given all previous amino acids. So there are certain patterns repeating for membrane-bound proteins which definitely differentiate them from all the others. And yes, of course, we also trained something on top of these embeddings. We did not only visualize them with t-SNE. So we have here now a fixed size input. Therefore, we could use a normal neural network in this case, because if you're sure that your vectors will always have the same size, which is the case here, then you can definitely use the neural network. We do a single hidden layer transformation to 32 features. We apply dropout, a rectified linear unit, also batch norm. And yes, I know that dropout and batch norm are not really frequently used together. This was rather a result of plug and play and see what the results are. And yeah, it improved slightly by simply adding the batch norm, even if it's counterintuitive, but it worked. So I just left it, kept it in there. And then we did again a multitask prediction. We predicted whether a given protein is membrane bound or not. And we also had labels for the same data set for in which subcellular localization these proteins are. So we knew whether it's in the nucleus, for example, or in the mitochondria. So first of all, let's talk again about membrane versus soluble. We applied the same workflow, so we embedded our input sequence via the pre-trained ELMO. We got the embedding matrix L by 1024, yes?
Just one question regarding your architecture. Why do you use the batch norm after dropout and reload?
Yeah, I tried all combinations, and this was basically the one which worked best. So I agree, this is counterintuitive, as I've said. I cannot explain you why it's working better.
Because like the dropout should really mess up your batch norm.
Yeah. This is also what.
Increase the dropout, that would have been better probably.
Sorry?
For that, did you try increasing the dropout here?
Yeah, that's what I said. So I really, because this whole thingy trained in like 5 seconds. So this was one of the few examples where you could really plug and play with the architecture and just see the effect. And this was also my assumption that this should completely mess up everything if I do this combination. But no, apparently not. This was interesting to me, and I tried to read on this on the internet, and I found several cases where people actually said that they observed something similar. Others said that they observed exactly the opposite. So it is difficult. It's just an empirical result. I just tried different combinations, and it slightly improved the performance. But yes, I agree. It's counterintuitive.
You like standardize your input values, maybe not? And then like, because you maybe had that initialization or something, that's why it didn't work.
So the input should be normalized. So the output of ELMO, so these embedding features are already normalized. So what you get out here is, so this is just a toy example obviously, but the values are actually already normalized because they are intended to be used before some other architecture. So the only use case for these embeddings is that you use them basically as input for something else, and therefore it made sense to already normalize them. And so that's why the embeddings are definitely already normalized at this point. But this is something, we published the pre-trained model, the data set is also published. This is something which, if you have experience with CNNs, can be done in like 2 hours of work, I would say. Feel free to just plug and play and experiment by yourself. But I agree, so that's why I've mentioned it. So I know that this is counterintuitive.
I'm just 90% sure like substantialization was done wrong, or maybe the weight initialization was done wrong. That's why batch norm improves here. But so we might look at that, but maybe not. Maybe it's
yeah, no, I agree. So I tried to look into this because I was equally surprised. But yeah, again, this is just work published a few weeks ago, so under heavy construction, I would say. Okay, so yes, we also trained again this very weird looking architecture, and so now if we now basically extract this hidden layer or the representation of this hidden layer given some input, some protein as an input, and then we visualize these 32 hidden, so this feature of size 32 in, with t-SNE again, then you already see that the clusters are much better separated. So you see definitely here is now a cluster for the membrane-bound proteins going down here a little bit, but up here you see nearly no pollution anymore, as well as here. So you always have some noise in there, but in general you see obviously that training of this network improved the interpretation of a given protein, whether it's membrane-bound or soluble. And as I've said, we did this not only for membrane-bound versus soluble, but we did it also for 10 different subcellular localizations written down here. In square brackets, you always see the number of samples for these classes, so that you see that we are also fighting with a heavy class imbalance here. So for nucleus, we have 3K proteins, and for the peroxisome, we have 73. So you can imagine that there will be a huge difference in the performance for nucleus and peroxisome. And this is the t-SNE representation of the raw Elmo embeddings again, so no pre-training, and you can also already see again here some clusters. It is a lot more blurry, so I would not argue that there is a super strong signal in there. So you see a lot of pollution, but you also see some nice clusters which are well separated from the rest. And again, this is without any notion or training on subcellular localization. And then we trained the same architecture, and you see, as expected, the result improves tremendously. Now you see here some nice clusters for extracellular, here for mitochondria, and here for endoplasmatic reticulum, for example. And this is, by the way, also a possibility to look into what your neural network actually learned during training, so to somehow get a notion of what is going on in these layers, so what the representation which are learned in these layers can look like. And now let's just again compare to other methods out there. So here is log32. I think you might have heard in one of the lectures also about this method, because it was also published in ROSLab. So this is a hierarchical tree structure of several LSTMs, which use also further pre-processed type of PSSM to make a prediction of 18 subcellular localization classes. And there are just several other subcellular localization predictors, and we compare to DeepLog, which is currently considered as one of the state-of-the-art methods, and they reach accuracy of 78% for this 10-class prediction problem, and we reach 68%. As a comparison, Seqvec is using the Word2Vec algorithm, so this algorithm which has always the same feature representation for Apple, for example, no matter what the context looks like. So here between this bar and this bar, you can see the difference between language models which are contextualized, so our approach, Seqvec, and language models which are non-contextualized, Seqvec, which are over here. For Seqvec, we applied the same idea, we just averaged over the embeddings of a protein. Okay. Now just finally, a short introduction of the underlying architecture of NetSurf P2, because this might actually explain a huge part of the performance difference between our approach and their approach, because again, we used a very simple CNN model, which had only few free parameters, versus their approach and their architecture, which had like 33 million free parameters. So what they use down here, again, the amino acid sequence as one hot encoding, so this gives you 20 features. They also used hidden Markov model profiles, giving you 30 features, so 20 for the replacement probabilities, but also probabilities indicating gaps and all that stuff. And so they had an input which had size n by 50, so they replaced basically what I wrote always as L with n, so they have an input of n times 50 as an input, and then they create a CNN which has a window size of 129 over this versus our 7, and this is, they train 32 of these kernels. On top of this, they apply a CNN with a window size of 257. They do this 32 times, so you already see that this is getting huge. Here they are concatenating basically basically the output of this CNN layer and their original input. So this is similar to the idea of residual networks, where you allow skip connections between different blocks in the architecture to allow easier backflow of the gradients on the one hand, but also to allow the network to actually choose whether this transformation is helpful for the current residue. So this is just a concatenation of the output of this layer and the original input, and then they train a bidirectional LSTM on this, again 1024 hidden layers. They do this in a bidirectional way, so they get 2 times 1024, they concatenate this again, and then they feed it in again 2 bidirectional LSTMs with again hidden layer size of 1024, and then they do this multitask prediction style. So they are also predicting 8 classes in secondary structure. So the accuracy measured over these 8 classes is called Q8. For 3 classes it's called Q3. RSA is the relative solvent accessibility of a protein or a residue in a protein. So it just indicates whether it's rather on the surface or in the core. Phi and psi angles just indicate the orientation of your side chain or the backbone and the neighboring residues, and disorder again is just whether it's disordered or not. And down here you see exemplary PDB structure where they have colored their predictions accordingly. So they have colored all predicted helices in yellow and all predicted sheets in like purple, and what they want to show is basically just that they perfectly hit the true labels. So this is, I guess, a cherry-picked example here. And what they show up here is visualization of their relative solvent accessibility prediction for the same protein, where blue indicates that the residue is rather buried and red indicates that the residue is rather on the surface. And again, yeah, this is just a cherry-picked example to somehow visualize the predictions.
What's the reason for using these huge filter sizes?
So this is something which you would best ask the authors. I was also super surprised. So I think they are hoping to just, by extending the neighboring windows, to introduce a notion for long-range dependencies. So the problem is that, is there a good example in here? So you can imagine that 2 residues in a protein sequence are quite separated in sequence space, but as soon as the protein folds, they get close in 3D space. Even though they are far away in the sequence space, upon folding they could get in contact.
But for example, in computer vision you would use several layers.
Exactly.
Smaller filters, so you also increase the interceptive field.
So actually yesterday I was about to prepare these slides, and I wanted to include another method which does exactly this. So one way would be to do it in this way. So I do not want to introduce any notion here, but I would agree, I would also use this approach. I would not say that this is really clever to introduce these large CNNs. And the other approach really used many stacked residual connection blocks, and again, this also allows you to train way deeper layers while still having these long-range dependencies in there. And this new approach, so this was published even after the NetSurfP story, a few weeks after them, and they even claim to reach 0.5% more performance like this approach, something like this. So it seems apparently to work better, but it also seems to does not make a tremendous change. Also a little bit surprising to me. I would have assumed that the residual blocks work way better.